financially, job-related, or perhaps loss of hours. And some of us perhaps have just been impacted on our just personal well-being and our mental health. But all of us have been impacted one way or another. And, we're looking at the numbers, and of course we think we're starting to come down from the peak of the second wave, and that is encouraging. But it's important to remember that even here in New Jersey, we've lost over 20,000 people have lost their lives to the pandemic. Even in this country to this day, we're losing about 100,000 we have about 100,000 new cases every single day in this country. And it won't be long, unfortunately, that we will have about 500,000 deaths across the country of our fellow Americans to this pandemic. So it's no surprise that vaccines are a hot topic. All of us are looking to vaccines as one way to hopefully emerge from this pandemic. Here in New Jersey, we've now administered 1.1 or over 1.1 million doses. Nearly 250,000 people now have had their second dose. So there is progress and perhaps hopefully some hope on the way. And so we here as Mount Olive Democrats, we advocate for a healthy and prosperous Mount Olive and certainly the United States of America. And that's why we're pleased to be able to present this nonpartisan event open to the entire community on vaccines featuring Dr. Sean O'Connell. But before I introduce Sean, I just wanted to say that all of you will notice you are on mute. This event will be recorded. We will be posting the recording on our website shortly after the event. Sean does have a couple questions that people submitted in advance, so thank you for those who submitted those. We will be monitoring the chat box, so if you've used Zoom before, you're familiar with the chat. If not, you'll see it on the bottom of your screen. And if you have questions or comments during the event, please feel free to enter in a comment, making sure that your chat goes to everyone so it's all visible. And then Irene, myself, or Dr. O'Connell will be able to monitor the chat. We will try to end by about 8:15. So Sean has a lot of excellent material to cover, and then hopefully we'll have some time for questions towards the end. So with that being said, it is my pleasure to read an introduction here. Dr. Sean O'Connell is a resident of Mount Olive. He is a seasoned healthcare professional, now with 35 years. Is that possible, Sean? 35 years experience in immunology, cell biology, regenerative medicine, and tissue repair. He is the founder and president of O'Connell Consultants. Dr. O'Connell has served as an interim management or strategic consultant to numerous venture capital, pharmaceutical, biotechnology, medical device, and tissue processing companies, focusing on operations, preclinical and clinical development, commercial opportunity assessment, product and market development strategy. His clients have spanned early stage startup through large cap companies. Dr. O'Connell is chief scientist in Eagle Research, an international catalyst partnership commercializing immune cell therapies, regenerative medicine therapies, medical interventions, and healthcare centers. Dr. O'Connell received his PhD in cancer immunology and molecular biology from Rutgers University and the University of Medicine and Dentistry of New Jersey at New Brunswick, now known as the Rutgers Medical School. He's had additional postgraduate work, including postdoctoral fellowships at the Department of Toxicology, Rutgers University, and the Center for Advanced Biotechnology and Medicine. He served as an assistant professor in the Department of Surgery at R.W. Johnson Medical School, where he was the head of surgical oncology and flow cytometry in immunology laboratories. Dr. O'Connell has authored over 65 peer-reviewed publications and abstracts, and currently holds a position of adjunct professor in the Department of Vascular Surgery, Englewood Hospital and Medical Center, Mount Sinai School of Medicine. And everyone has a fun fact, and the fun fact with Dr. O'Connell is he's an avid guitarist. He has a studio and a soundstage in his house. He loves the Strat Plus. I don't think we're going to hear it tonight, but we are going to hear everything about vaccines and treatments, making sense of it all. Dr. O'Connell, thank you so much for being with us tonight.
Well, the first thing I have to tell you is Andy's to be commended for getting through that with a plum, and that is impressive. Yeah, so as much as it'd be nice to get to know one another, I think we have a lot of material to get through today, and some of it's going to be familiar to some people, other of it's going to be new. We have a very diverse audience out there, so I'm going to try and go at a pace where we at least get through the essentials, and then open it up to questions. If it's a simple question, we're going to try and answer it tonight. If it's an involved question, we're going to take your name, your email address, and we'll get back to you with a thorough answer. Let's see if I can now work this. All right. So how do I advance my slide? That's how we advance the slide. All right, so give me a second here. All right, so what are we going to talk about today? We're going to talk about the COVID-19 vaccines, those that are currently available and some of the ones that are coming up. We're going to talk about a little bit how they were developed, what they are, how do they work, and this is the whole process of vaccination. How do they differ from conventional vaccines that we all are familiar with, like the flu, the MMR, smallpox vaccines? And importantly, how are they tested, both for safety and for efficacy? All right. In addition, we're going to spend some time on the new corona variants. These have caused a lot of consternation, a lot of comment, worry. We ask the question, how well do the current vaccines work against those variants? How does that differ from their normal response? Then we're going to talk a little bit about vaccine distribution. I'm sure something that we're all very interested in. Who's eligible? What's the order? When can we expect, what kind of timeline are we going to have as far as receiving these vaccines? And then finally, I'm going to end with a little discussion about safety, normal side effects of vaccination and the more serious side effects of vaccination, anaphylactic reactions that occasionally, rarely do occur, and how can we mitigate against that? All right, so getting on to vaccine and how they work and what their efficacy is. So vaccines basically work through the process of vaccination. And in doing that, what we're talking about is taking a preparation and stimulating a person's immune system against an organism. It can be the bacteria, can be protists or worms and the like. Most commonly, they're viruses. And we're going to talk about a number of the new vaccines that were developed against COVID. These include the RNA, DNA vaccines, viral vector vaccines, protein vaccines, and the concept of a cassette platform. Each one of these new vaccines represents a cassette that can be changed and updated very quickly. We'll talk about that later. And then finally, we're going to talk about, as I said, about the efficacy. How well do they work at protecting us against the infections? All right, so first of all, we have to get back to brass tacks, basics. What is vaccination? And it's the administration of a vaccine to stimulate the immune system to protect against diseases, usually an infectious virus or bacteria or other organisms. Traditional vaccines were generally made by taking the organism, we'll say a virus, and either weakening it through a number of ways or killing it chemically, and then administering to the patient to confer protection without giving them the disease. This was later extended to using purified proteins and toxins from these organisms and using them as the basis of the vaccine. These vaccines can either be given in one dose or multiple doses. Multiple doses usually have a boosting effect on stimulating the immune system. They're often by injection, but they also can be taken orally, like the Sabin vaccine, or nasal vaccines are starting to come in, nasal sprays. In the process of vaccination, the body recognizes the vaccine itself, the immunogen, as foreign, mounts a response, and creates memory cells. These are very important because immunologic memory is going to allow us to be protected going forward against the organism, in this case, COVID. And so that after we're vaccinated, also, if we've survived COVID, when we are confronted with the organism again, our immune system will remember that and mount an extremely rapid response such that you wouldn't even know it happened. This response is basically composed of two elements. The body makes antibodies in response to the organism, in this case, COVID-19. And it also makes T cells. These T cells basically are how the body recognizes virally infected cells and kills them. Antibodies, on the other hand, recognize the free-floating viruses, bind them, and destroy them. The end result of this is that when a sufficiently large percentage of the population is vaccinated, we get herd immunity. What does that mean? That means at some point we have enough people who are resistant to the disease that the disease can no longer get a foothold and basically burns itself out. And you'll hear a lot of talk about herd immunity. Currently, right now, the estimates on herd immunity for COVID in the United States are about 70%, 75%, according to Fauci. That means we got to get 75% of the people either had the disease or are vaccinated to achieve this halting of the spread of the virus in the US. All right, what are some of the vaccine types that we're going to be seeing now that are currently available? Well, the most prominent one that's getting a lot of the press are the genetic vaccines, particularly the RNA vaccines, of which the most common examples right now are the vaccine produced by Pfizer, as well as the vaccine produced by Moderna. There are DNA vaccines that are in development, but they haven't come as far as either the Pfizer or the Moderna. And they're listed here, but I'm not going to talk too much about them because you're unlikely to run into them until maybe the latter part of this year. If someone's interested more, I can talk later about it or send them something. Another major type of vaccine are the viral vector vaccines. These kind of vaccines are vaccines that contain viruses that are specifically engineered to carry the coronavirus genes to the person being vaccinated. Some of these viral vectors, vaccines enter the cells and cause them to make proteins like the RNA vaccines, which are then expressed, the body sees it, makes a response to it. Other types of viral vectors grow slowly, carrying the coronavirus protein with them on the surface so the immune system can see it. Examples of the viral vector vaccine include the J&J vaccine, which I think just got emergency use approval, the AstraZeneca vaccine, and the Russian Sputnik 5 vaccine. Those are all viral vector vaccines. And then you have protein-based ones. The most prominent one is Novavax, which is now starting to get a lot of airtime, and it's a good vaccine. In this case, what you're doing is you're taking proteins, but no genetic material, and you're mixing them with agents called adjuvants that are going to stimulate the immune system. In the case of Novavax, they actually mix it with nanoparticles, and they administer that. These are fragments of the protein on these little particles. These vaccines seem to be highly effective. I'll talk more about Novavax in a little bit. And then finally, you have groups that are making conventional vaccines. Either they're trying to inactivate COVID or attenuate it by growing it in tissue culture. Problem is, there aren't any being developed in the United States. You see them mostly in China and Asia. They have lower efficacy, where the other vaccines are like 70-some% or higher, 95% in the case of the mRNA vaccines. These vaccines typically have an effective rate of about 50%. In other words, kind of like our seasonal flu viruses, and it's basically made in a similar fashion. And so they don't have the effectiveness. The good news is they're relatively simple to make and they're cheap. So you see them outside the United States. All right. So the vaccines we're going to look at today is mostly the Pfizer and BioNTech, which is made with a collaboration of Pfizer and a biotech company in Germany. It has emergency use authorization in the United States. The Moderna, which comes from New York, right here, right local, that's got an EUA. The AstraZeneca that was developed as a collaboration between the pharmaceutical company AstraZeneca and Oxford University in England. Its EUA is pending. Then Johnson & Johnson, which is a collaboration between Johnson & Johnson, Janssen Medical, and the National Institutes for Allergy and Infectious Disease. That one is imminently going to get approval if it hasn't gotten so already. And then finally, as I said, Novavax, which is the most recent one on the scene. All 5 of these have been through phase 3. So they've all completed their phase 3 study, and they're currently under review. But of the 5, 3, at least I know of, have emergency use, and all 5 will probably eventually have emergency use, allowing their introduction within a short period of time, within a month. But it should be noted that there are over 144 different vaccine preps currently in development, so it's hard to keep them straight. All right, let's quickly talk about the big 5. All right, the Pfizer vaccine, which is the most common here, with Moderna number 2, is an RNA vaccine that requires 2 doses. The doses are spread out by 3 weeks, 21 days. It contains RNA, which is the message from the gene that codes for the spike protein on the COVID. The spike protein is what allows the COVID virus to enter a target cell in the body. And so the way this vaccine works is you inject it intramuscularly, and they studied adults 16 years and older, not younger. And what happens is it's taken up by the muscle cells. The muscle cells read, if you will, the RNA and make the protein, part of the protein for the spike protein. And then that's expressed on the surface of the muscle cells. The muscle cells can then show it to the immune system, if you will. The immune system sees it and then makes a response, makes both an antibody response and a cellular response, which I say in the next one. This vaccine has turned out to be highly effective. It's against the parent strain, the COVID-19 originally out of China. It's 95% effective. What does that mean? What do we mean by efficacy and effectiveness? What it means that is if you have patients in a trial, say you've got 100 in the treatment group, they all get the vaccine, 2 doses, and you have 100 that get a placebo, generally saline, salt solution, and they get 2 injections. After the second injection, a certain defined time, defined in the study, you ask the question, all right, how many in the treatment group contracted the disease and how many didn't, versus in the placebo control, how many contracted the disease and how many didn't. What this means then is that in the treatment group, if we had 100 patients, we only expect 5 of them to get COVID. This is as opposed to the control group where you see most of the patients would be susceptible and have contracted the disease. That's what we mean by efficacy, the ability of the vaccine to prevent infection. It doesn't talk about anything else. It doesn't talk about the severity of the disease. It doesn't talk about transmissibility. It doesn't talk about inflammatory responses that may affect organs. It's just a measure of how well the virus protects you against infections. In the Pfizer vaccine, over 40,000 people were tested. There were no significant adverse events in those 40,000 in the phase 3. The big drawback, though, with the Pfizer vaccine is it needs to be kept really cold. minus 70 degrees C, which translates to about minus 94 Fahrenheit. That's pretty darn cold. So you need special shippers, special freezers to maintain this. And that makes distribution of this vaccine, an issue, a problem. Quickly, the Moderna is a very similar virus. It's an mRNA vaccine against COVID. It's 2 doses. They're 28 days apart. Again, muscle injection. The only difference is in the Moderna, they started at 18-year-olds and went older. And its efficacy is almost the same, 94.5% efficacious at preventing COVID when measured 7 days after the second dose. And I should indicate that both the Pfizer and the Moderna virus work pretty much the same against the UK strain, which I'll talk about later. It was tested in 30,000 patients. It's a little better storage. You can store it for a month in the refrigerator and 6 months in your regular freezer. You don't need the ultra cold. This is our regular minus 4 degrees Fahrenheit free refrigerator freezer. It has better storage capability than the Pfizer, and hence it's easier to distribute. Now we get into the viral vector vaccines. The first one that was introduced was the AstraZeneca. It's a 2-dose vaccine, but they separated it by 12 weeks, which I thought was a little excessive. And it's made, the basis of it is an adenovirus that originally came from chimpanzees. They basically re-engineered it. It's no longer infectious, but it can be used to carry the genetic information, in particular the S protein. It's administered through muscle injection, and it's got pretty good efficacy. It's about 82% after the second dose. Its big drawback, though, is it's shown not to be very effective in either the South African or the Brazilian strain. And the company is racing to come up with a variant that would be directed against those two strains, the South African and the Brazilian. Again, it's got very good side effect profile, no severe side effects seen in any age group. However, one of the big drawbacks of their trials was they didn't really look at it in older patients. They kind of sort of stopped at 55 years of age with just a handful of older patients. Again, it's stable in a refrigerator for 6 months, making it very easy to distribute. All right, the Janssen drug is, I think, a very promising one. It's currently being released as a single-dose drug, though they're trying a 2-dose formulation as well. It uses a human adenovirus as the carrier. Again, they're using genetic information for the COVID spike protein. Muscle injection, they looked at patients 18 years and older, and it's about 72% effective, but it's very effective at preventing severe disease. It has similar results in the UK strain, but again, reduced efficacy in the South African and Brazilian strains. It was tested in 45,000 patients, and you can store it for up to 2 years in a regular freezer and up to 3 months in the refrigerator. So again, it's a stable vaccine. And then finally, Novavax. This is a protein. It's no genetic information, no virus. It's a purified protein. They use recombinant technology to get the viral proteins, so they're not infectious. They can't cause infection, but they induce a very good response. 2 doses, 3 weeks apart. Again, muscle injection and almost 90% effectiveness. However, again, you see a drop when you look at the South African strain. 30,000 patients in the US, 15,000 patients in England, and you can store it in a refrigerator. So in a nutshell, I apologize for all the detail, but that's the 5 lead vaccines that we have now. Now, a question has come up in the press, and I hear it a lot, is didn't we move too fast to make these vaccines? Have they been rushed? Did they cut corners, or did they use the regular process? And they say, and there's some basis if you compare it to the older conventional vaccines, because they usually took years to develop. Up until COVID, the quickest vaccine on record was the Mumps vaccine of the MMR, and it took 4 years to develop. The answer to the question is that both the RNA vaccines and the viral vector vaccines had already existed as platforms for many years. They've literally been studied for decades and have been honed. I know about the Oxford vaccine. I know about the Moderna vaccine, and I'm aware of the Janssen vaccine, the Adeno 24 vaccine. These have been around for a long time and have been used to make vaccines against diseases like Ebola, Zika, dengue fever, and even HIV, though we're still waiting for a successful version of that one. But my point is they've been around for a while. And as I said in the beginning of the talk, they exist as cassettes, by which I mean there are platforms that if you have a new DNA sequence or RNA sequence from a new variant or a new virus for that matter, inserting that sequence into the cassette can give rise to a brand new vaccine in a very short amount of time. And that's what I say in the last bullet here. Once the virus sequence was available online in January 19th of last year, 2020, Dr. Sarah Gilbert at Oxford University had a working vaccine prototype by the middle of the next week, which later would become the AstraZeneca vaccine. So this is a new chapter, a new era, if you will, in vaccine technology. We've not had this before, not commercially anyway, that you have these platforms that are rapidly adapted to anything for which we have the sequence, either the RNA or the DNA sequence. So that means for these variants, we're going to be able to come up with new booster vaccinations that are specifically designed to the variants, and we're going to be able to do it quickly. All right. So let's talk about the variants. This is a map of SARS-CoV-2, our now old friend that's been causing all the pain for the last year. All right, the sequence was published by the Chinese January of last year. And as I said, all the current vaccines that we're looking at are quite effective against it, much more so than historical vaccines. However, what you need to know about these kind of viruses, these RNA viruses, is they mutate very fast. As they infect someone and they get into the cells and they direct the cells to make lots of new virus, mistakes can happen. These mistakes are called mutations. You see this with a lot of other viruses. You see this with influenza. You see it with HIV. Organisms that use DNA don't have this problem as much. They can actually check or proofread when they divide. Not so the RNA viruses, and a lot of mistakes get made. Well, some of the mistakes, you can track these mistakes. You can actually figure out viral lineages and define family trees, if you will. If a group of viruses starts to show the same mutations, they call that a variant. Now, most of the mutations have little or no effect. As I say, they occur all the time. If they have a adverse effect, the virus is quickly lost. That mutation just drops out. However, if you get enough mutations that accumulate that confer some benefit on the virus, that gives rise to what we call a strain, a variant strain. COVID is caused by a coronavirus strain called SARS-CoV-2. And as I said, the current vaccines are quite effective against it. However, we now are seeing new variants, which have a number of mutations, and that's listed by the red dots, the red pins there, if you will. Those are mutations. This is the UK strain, B117. It doesn't cause you to get sicker, but it is so much more infective, so much more contagious, if you will, than the standard COVID. How much? About 40 to 50% more contagious. So it's going to cause more disease. A given individual exposed to this is more likely to be infected than the parent strain. The good news is, as I said, all the vaccines are very effective against it. And that's very important because right now the strain is in the United States, and every 10 days it doubles in its occurrence. It will most likely become the dominant strain in the United States within the next 2 to 3 months. All right. Now for the more problematic ones. The South African strain B1351 and the very similar Brazilian strain have different mutations. And they have mutations in the spike protein that do render the current vaccines not as effective. The Novavax was up to 90%. Against this strain, it's 49. The J&J was up in the high 80s. It's now down to 57. The AstraZeneca was up in the high 80s. It's now down to 10. And even the mRNA viruses drop about 30 to 40% in efficacy when confronted with this strain. The good news is all these vaccines, while they may not prevent infection, do prevent the development of severe disease. This is very important. You hear this all the time with flu, that even if the flu isn't so good this year and it doesn't cover most of the strains, you should take it anyway. Why? Because it still seems to lessen the severity of the disease. In the case of flu, it prevents pneumonia. And in this case, it seems to do the same thing. You don't get pneumonia. You don't need to go into the ICU. You don't have to go on a ventilator. So there's a benefit from taking any of these vaccines when it comes to blocking the development of severe COVID-19. All right. As I said, we have a cassette platform. And using that cassette platform, these variants, as you saw by these maps, we already have the sequences. We have the RNA sequences. All the 5 companies already have variants that are testing right now. The good news on that is that all they have to do is A, show that it works against the variants. These are the vaccine companies, and B, that it's still safe like the parent vaccine. What this means is that once they've got a booster, for example, against the Brazilian strain that's ready to go, it's only going to take about 6 to 8 weeks to get approval once it's out. So it won't take nearly as long as the original vaccine. That's the good news. All right, let's talk a little bit about vaccine distribution. To date, 33 million people roughly have received 1 dose, with almost 10 million fully vaccinated. This is data from the CDC. This rollout, though, varies significantly from state to state. We're going to talk mostly about New Jersey and the surrounding area. In New Jersey, we're up to about 10% have received 1 dose, and about 3% now have received 2 doses. However, the demand is greatly outstripping the supply. This was particular, as you'll see in a minute, when they changed the criteria back in mid-December to basically open it up to anybody over 65 and anybody 16 to 64 who had comorbid conditions. When they did that, in other words, you move beyond frontline healthcare workers, you move beyond essential workers like police and firemen, when you move beyond people that were in nursing homes, you then increase the demand so much that manufacturing just couldn't keep up. So we need to improve this. The Biden administration started with a pledge of 100 million doses in 100 days, but they now admit that they can make more, and they're going to have to make more. Based on the current estimates of herd immunity in the United States, that means we need to vaccinate between 70 and 90% of the patient. We need to do this in order to stop the infection, to get a resistance enough to slow down this community transmission. The time to that herd immunity is going to depend on how fast we get the vaccines out, as well as the degree and the extent of the vaccines, which are very high. So what are we talking about? Right now, we have about 920,000 daily doses. At that rate, we'll hit the 50% coverage point by early July, and we won't hit the Dr. Fauci's mark for herd immunity until late September. We need to do that quicker. So what's happening is the supply is improving, the distribution is improving, and so we think that the rollout will get better. There's not only supply itself, but we also have to get better at the logistics of administration. And this includes central control, central coordination, I think out of Washington and then through the individual state centers, but it has to be centralized And it really has to be coordinated. Doing that, we will be able to shave quite a bit off of this September mark where we start to encroach on herd immunity. This is the distinction that Dr. Fauci likes to talk about by saying, well, there are vaccines and they can be very good, but then there's vaccination. As a country, we really need to focus on the vaccination now. We got good vaccines. We know how to use them. We just got to get them out there to everybody. All right, who's eligible? Well, the initial guidelines were, as you see in the first bullet, you had phase 1A, 1B, so frontline healthcare workers, personnel, long-term care residents, and faculty. Then you had 1B, which are first responders and essential workers. Phase 2 was older people with comorbid conditions like myself. And then phase 3 was everybody else. That all changed on December 20th. Now, 1B is really everybody 65 and older, or people 16 to 84, or 64, that's a typo there, 64 with a comorbid condition. And I'll show you what those comorbid conditions are for New Jersey in a minute. What does this mean for Jersey? It means 65 plus high-risk individuals and some essential workers, but not, interestingly, teachers and grocery personnel. They're trying to work on that. I think they need to open it up to the teachers because we need to get the kids back in school. There's no question about it, but we have to do it safely. And that means getting the teachers vaccinated. All right, here are the risk conditions. So if you're between 16 and 64 for the Pfizer, or 18 and 64 for the Moderna, and you have either cancer, a history of cancer, chronic kidney disease, chronic obstructive pulmonary disease, Down syndrome, significant heart conditions, obesity, and especially severe obesity, sickle cell, smoking, or type 2 diabetes, you are eligible for the vaccine right now. But the issue is we don't have the supply yet. We need to get the supply going. All right, so last, I'm going to close with a little description of safety. How is it done? Well, essentially, all these vaccines, like pharmaceutical, like drugs, go through a set of tests, phases of tests. In preclinical tests, new vaccine is tested on cells and see if it can stop the virus, in this case, using cells in the petri dish that are susceptible to the COVID. And then once you show that it works against cells and can protect cells, you move to animals, usually mice, monkeys, sometimes other ones. And you can see, the first thing you see is, does my vaccine produce an immune response specific to the virus? And then you say, all right, if I challenge the animal with the virus, will it protect them? If the answer to those is yes, and there's no gross adverse events in the animals, the safety looks good in the animals, you move to phase 1 clinical trials. And in phase 1, the vaccine is given to a small number of patients, of volunteers, and you test both safety and dosage. You want to see where you are. And in this case, specifically, you want to see, do I get a good immune response using this vaccine? If the answer is yes, you go on to expanded trials. Now you're giving the vaccine to hundreds of people, split into groups. In other words, say we stratify. We have children, maybe, unfortunately, not often enough, or we have elderly people, or we have people that have chronic conditions, and we see if the vaccine works in them. In addition, in phase 2, we look at the safety. Now, these phases can sometimes be combined. What you'll often see is phase 2 combined with phase 3. This is your randomized controlled efficacy trial. It has to be done on multiple sites. It has to involve thousands of patients. And again, it's very strict. It's controlled. So that means half of the patients are going to get a placebo and half of them are going to get the actual vaccine itself. And the whole idea is to determine the efficacy rate. That's the primary endpoint. As I said earlier, what percentage of the people receiving the vaccine are protected against the virus as opposed to the placebo group? And that determines the rate. That figure, that differential, is your efficacy rate. In addition, you're going to look for relatively rare side effects. Now you're treating tens of thousands of patients that we see. However, really rare side effects will still not necessarily be seen in phase 3. Based on that data, the vaccines can apply for limited approval, which is exactly what has happened in Pfizer, the Moderna, and the J&J. Full approval usually takes a much more thorough review of all the data, phase 1, 2, and 3, and the preclinical stuff. No vaccine currently has full approval in the United States, though in Europe, I mean, England, I know they've approved a couple of them now. And it's really a matter of just slogging through the data and making sure they didn't miss anything. But based on the phase 3 data, you can pretty much make a determination whether you can give an emergency use authorization or not. Now, one thing they're not talking about that's very important is post-approval surveillance. This is an ongoing process that occurs for any vaccine as well as any drug. And the idea is that you continue to look for side effects, because some of the side effects are so rare, like anything 1 in 100,000 or greater, that you won't pick them up in your phase 3 trials. It's very unlikely you'll see them, but you still want to know about them. So the FDA has a number of post-surveillance programs going that's going to follow these vaccines once they get out into the population, and you start treating hundreds of thousands and millions of patients, and they're going to still look to see if anything is popping up there. So safety monitoring doesn't stop with approval. All right. What are we talking about by side effects? Well, there's 2 kinds. There's common side effects associated with the virus, and these include, they're mostly minor. They're fever, sore arms, chills, flushing, headaches, muscle pain, joint pains, nausea, swollen glands. But the thing about it, fatigue, but the thing about that constellation of effects, which are common, they generally resolve within 24 hours, maybe 2 days. However, you have severe reactions that do occur very rarely. How rarely? Well, a conventional vaccine like the measles, mumps, rubella, or the polio, or the smallpox vaccine, you don't see a severe reaction until you get to about 1 in a million, a frequency of one event in a million administrations. What are they for our new vaccines? In the Pfizer, the most recent report I heard last week was that they're seeing about 6 severe adverse events in a million. And in the Moderna, about 2 in 2 million. So they're not common. They're not common at all. What do these severe reactions look like? Well, they're rapid onset. They occur within minutes to hours. They start off generally as an itchy rash, throat or tongue swelling, shortness of breath, lightheadedness, vomiting, but importantly, often low blood pressure, crashing blood pressure. And yes, they can be life-threatening. They are very similar to bee sting reaction or reactions to penicillin allergies or peanuts. They're significant. How do you treat them? Just like you do with bee stings and peanut allergies and sulfa allergies. You mostly use epinephrine, an EpiPen, as well as providing supportive care. With those interventions, they are all reversible and the symptoms go away very quickly, but you do have to be prepared for them. There is another kind of severe vaccine reaction called the Guillain-Barré syndrome. So far, it's not been detected in any of the COVID vaccines. However, it was a big thing a number of years ago when they had the first swine flu vaccine back in '76. There was something very funny about that influenza vaccine, and they had a rash of Guillain-Barré syndromes, several thousand. But as I said, so far, this syndrome has not been seen in COVID vaccines at all, none. So what kind of precautions can you take? Well, if you are an individual that has has a history of serious allergies, like bee sting allergies or peanut, other food allergies, or allergies to medication, the first thing you want to do is, before you get your vaccine, once you know you're going to get it, either call ahead or tell them when you arrive at the clinic that you are such an individual, that you do have a history of of allergic reactions, significant allergic reactions, and they need to be ready for it. So you make sure then that the clinic is set up to handle these severe reactions, that they have epinephrine, EpiPens, that they have supportive equipment. And so they then, oh boy, I want to go backwards. Sorry about that. That they're ready for it. And then there's one further thing you can do. The normal regulations state that you should wait 15 minutes after receiving your vaccine. If you think you have a history at all of such an allergic reaction, you should tell them that, can I hang out for 30 minutes or more? Just to make sure before you get, you're taken back, someone drove you there, or you're driving yourself back, not a good idea, that you're fine, that you really don't have a reaction. But taking these simple precautions basically can cover all of it. I am now at the what? I'm at the 45-minute point. Yeah, a little past that. And I've covered an awful lot of information. The information I have can be found in a couple of pretty readily available sources. The ones that I like to use is see, I have a list here. Yeah. The New York Times has their coronavirus briefing. The Washington Post has its coronavirus update. The Journal of American Medical Association has its COVID-19 update. Medscape has a COVID-19 update, and so does WebMD. The good news is all of those sources, you don't have to subscribe to the journal or the newspaper. You can just go on their website and sign up for the corona updates, and they're provided for free. And I highly recommend you doing that. I in particular found the New York Times to be the most comprehensive. The one from the American Medical Association, it's a little heavy. The Medscape one was also a very good one. And with that, I'm going to stop and take questions, but I thank you so much for your patience. Thank you kindly indeed.
Sean, thank you, sir. That was absolutely outstanding. We had two questions that came in advance. One was someone asked a question regarding if a cancer survivor and has celiac disease, and wondering if it was safe for the person to get the vaccine, and if so, if you had a recommendation on which one.
All right. The latter, as an immunologist, I would say I would go with the mRNA. What this person has is a history of cancer and autoimmune disorders. All right, so what does that mean? Their immune system isn't 100%. So what's likely going to be the effect? The effect is likely going to be that the vaccine may not kick in quite as hard or as strong as it would in a healthy individual. But there's been no information on any of the studies that are out so far that immunocompromised individuals are at risk for a detrimental vaccine reaction. And going to the viral mRNA, think of what you're doing. You're taking essentially a viral message. It's the program. And you're going to inject it in the muscle. The muscle cells are going to take it up. They're going to read the message. They're going to make the protein so the immune system can see it. And as I said, for this individual, the only worry is that you may not quite have the efficacy that you would have in a healthy individual. So if it were up to me, I'd say, yes, they should get the vaccine. That's what the consensus is, especially out of CDC. But they should probably get I would go with the RNA viruses. Why? They work quickly and have the highest efficacy.
Very good. There was also another question. The person would like to know if they should be concerned about taking the vaccine if the person is allergic to either sulfa or ampicillin.
Sulfa and ampicillin are not directly related in their structure to the COVID vaccine, but this might be what I was just talking about as I ended my talk, a hypersensitive individual. There are individuals that have hair trigger immune responses, and they can develop allergies and severe allergies quickly. A drug allergy is one indication of that. If I were the case, I would take the precautions that I just told you. Tell them that you should still get the vaccinated. The disease is much worse. However, you should take precautions. Tell them in advance that you have allergies to medications, and make sure that they are equipped at that site to handle those adverse events. And then finally, they should watch a little longer just to make sure there isn't an adverse event.
Thank you, sir. And I see we have a question in the chat box. Are you able to see that, or would you like me to read
I'm
it?
trying to get it up. If you give me a second, I'm trying to see if I can get out of aha. All right, now let's see if there's the chat box. We're up and running. All right, so which question is this?
This would be from Prateep.
All right. New variants of the COVID can be addressed with a booster vaccine that accounts for the new mutations, exactly as I said. With adenoviral vectors, there's a risk that people will develop immunity against the vector. I have a very good point. Given the potential for an importance of controlling viral variants, should the government pull the plug on funding for adenoviral vectors and repurpose that money for good. This is one of those ones where I'd like to consult my experts and get back to you, but I can give you a seat of the pants response. One of the reasons why both the chimp adenovector that was developed in Oxford and the adeno 26, which was developed at Beth Israel, are used is because they don't seem to have this predilection to getting sensitized. Another common adenovirus that's been used forever to make all sorts of construct is adeno 5. The problem is most of the population has antibodies circulating to adeno 5, so it's not appropriate. I think I understand what you're talking about here is if you received, let's say you received the AstraZeneca, and you're now confronted with the Brazilian strain, would your body expect it to recognize as foreign the new booster and diminish its its effectiveness? And my only answer to that is we'd have to test. But I don't think you're going to see anybody going to pull funding from the viral vector initiatives and push them over to the mRNAs because of this theoretical potential. The fact is the adenoviral vectors work very well. They're very flexible, and you'd really have to show that you got a problem with essentially sensitization and loss of efficacy in order to even get anyone to buy this. Should people that have had COVID still get the vaccine? Absolutely. This one I can be pretty firm about. Especially in people who have had mild, asymptomatic or mild to moderate disease, the immunity they get from that disease is not nearly as strong as what seems to be conferred on them by in particular, mRNA vaccines. Much stronger immunity. And to a similar extent, Novavax, the J&J, and the AstraZeneca. What the FDA is recommending, not the FDA, CDC is recommending right now is you should wait 10 days after subsidence of symptoms of your COVID disease before you go in and you get vaccinated. But the book on all the experts, from Schaffner down in Vanderbilt to Cotes, in Baylor to Osterholm, Ossip, Tony Fauci, they're all saying, yeah, get the vaccine. They're that good. Just wait 10 days after your symptoms subside, especially if you had a mild or asymptomatic and your COVID PCR test lit up. You still should get the vaccine. Next one. What we got?
Hey, Sean, if you scroll to the very top, that way.
Oh, I apologize.
Yeah, a number of them came in early on, so if you kind of go to the top there.
All right, so we just had, Prateep, I think we just got your questions. Irene, do children need to be vaccinated? Oh, this is a great question. Do children need to be vaccinated to achieve herd immunity? Monstrous question. The answer is we don't know. We know that they, in general, but not always, don't get disease like we do, like adults do. We do know that they certainly can transmit the disease. So the question is, in order to stop community transmission, which is what we got, For example, here in Morristown, we got it. Morris County, we got it bad. We're about as hot as you're going to find in the nation right now in community transmission. Do we need to vaccinate children to stop this? The answer is we don't know, but the consensus is if we get the adults down to 16 vaccinated, you're going to present enough of the barrier that there is no reservoir within the children where the virus can hang out. That's at least the theory in what the book says. Do I think this is right? No, I think we should, each one of these companies should actually open up at least small pilot studies to look at vaccination in these children. And the problem is because of liability, other reasons, they stopped at 16 or at 18 years old. I think that was short-sighted. I'd have gone down to at least 5, maybe 10 years of age. And so you ask a good question, I don't have a real good answer for it. Will we have any choice as to what producers we get? Probably not. My recommendation is right now, given where we are and there's just extensive community transmission, grab the first of those 5 vaccines, grab the first one that comes around. The only one I've gotten a little cold on is the AstraZeneca. And the reason is they haven't been completely forthcoming. They didn't study it in older patients. They didn't look at a number of issues that the other vaccine makers did. And so I'm a little less keen on that one, but all of them show good efficacy against the strains that we currently have around here. So the first one that comes along, I get it. Is there any concern for safety, those companies which may use preservatives instead of co-temps? No. The idea here is the preservatives that are used in conventional vaccines are designed to stop other bugs from growing in them. So you hear like thimerosal, some of these very low concentration mercuric preservatives. They're designed so you can put the vaccine at room temperature on the shelf or in a refrigerator, and it's not going to get buggy. These vaccines do not have that problem. The reason with these vaccines is you need to make sure that the mRNA, in the case of the Pfizer and the Moderna, and the viral vectors, which has got protein and it's got, a number of different proteins and the RNA inside, that they do not break down, and they can quickly, before you can administer them. So it's very unlikely with any of the vaccines that I talked about today, with the possible exception of the Novavax, that you're going to see an attempt to try and treat them like conventional attenuated or heat-killed viruses and have long-term preparations that will have chemical preservatives in them. I don't think you're going to see that with these kind of viruses, these vaccines. They're just a different animal. All right. Now, other people are asking what kind of things. Are the side effects much worse in second vaccinations? This is a very good question. And with respect to those minor, yeah, you could get hit harder. Doesn't happen to everybody, but you could get a little harder. You could have more nausea, more fatigue, more headaches, muscle pain, joint pain in particular at the site, muscle pain at the injection site, joint pain. But again, you can treat them with analgesics like aspirin, ibuprofen, and Aleve, and they're going to be limited. They're going to persist for about 24 hours. However, they're also an indication that the darn thing is working, that your immune system has been marshaled and is cocked and loaded and ready to go. Are these the same as the severe adverse reactions? No, they're not. Not even close. They're in the minor one, and they resolve rapidly. And they, other than aspirin, they don't need any kind of medical intervention. All right, we've talked about the kids. We talked about cancer. The problem with children is we just don't know. All right, here's a good question. This is JJO. My mom is 92 and has chronic thrombocytopenia, which she gets a shot every week to keep her platelets up. Works very well. They drop very low if not medicated. I'm worried about her getting it. Really, it's going to help her. I have a friend that just turned 100. She's in Carefree, Arizona, and Carol Bowman, wonderful lady, and she just got her vaccine, both shots. She got the Moderna, and she's doing fine. She doesn't have thrombocytopenia, but she has a history of a couple of different kinds of cancers. I don't see how it could hurt. There's no indication these vaccines interfere with platelet function. And the big problem with chronic thrombocytopenia is you have a crashing platelet count, so your clotting is affected and your response to bacterial infections, particularly infected, and that's covered with the injections. I don't see how the vaccine is going to interfere with that. I've heard no reports about that. All right, anything else? We have lots of questions. If a person is not allowed to take a vaccine with a live virus, should they stay away from any of these vaccines? No, none of them have live viruses. So the answer is no, they shouldn't stay away. We are not in the United States using any current vaccine preps that are either attenuated viruses or heat killed viruses. We're just not doing that. We're using either nucleic acids, viral vectors, or purified proteins. So all of the vaccines that we talked about today available in the United States are fine. Is it safe for a breastfeeding mom to get vaccinated? This is a great question, and I'm not going to give my own opinion at all. What I'm going to refer you to is there is a researcher at Northwestern. Her name is Emily S. Miller, MD. She's the chief of obstetrics and gynecology at Northwestern Med. And she's on Biden's panel. And she just published a paper in American Journal of Clinical Pathology. And she makes an extremely cogent and potent case for vaccinating of women who are thinking they're going to get pregnant, women who are pregnant, or women who are breastfeeding. And her rationale goes like this: even though they didn't study the vaccine in the phase 3 trials in pregnant women, there were a number of women who entered the trial and got pregnant during it. There have been no reported excess adverse events, either miscarriages or malformations or troubles with the pregnancy. In addition, there have been 10,000 documented frontline healthcare workers who received the vaccine while they were pregnant. They also had apparently normal term pregnancies. With respect to breastfeeding, it is known that the virus cannot enter the milk, just as it can't get across the fetus into the placenta. However, the antibodies do cross the placenta and do get into milk and do confirm immune resistance to the fetus or the newborn. This has been known forever and ever and ever. And it's well understood in immunology. So what Dr. Miller's saying, not me, but what Dr. Miller's saying is you should consult with your GP and your gynecologist, obstetrician. But Dr. Miller is very adamant. She said that the risk is far outweighed by the risk of the disease itself. And one of the things that you have to know about pregnancy is that pregnancy is associated with a limited but definite immunosuppression. Got to carry this young child who's got a different genetic background than you do, if you're the mother. And so there's a selective immunosuppression the body undergoes during pregnancy that is reflected in a reduced resistance to infection. And infected mothers get hit by COVID real hard. So I would tend to agree with Dr. Miller. And if you want if you want that report, I can get it for the people who need it. But this is what Dr. Miller said. This was on, she did an interview with Sanjay Gupta on CNN. And then I went and I dug up, a report, synopsis of a report. And she's quite adamant. This is a mother who's also chief of obstetrics and gynecology at Northwestern, and she thinks it's probably not a bad idea. So that's all I'll say about that. Anything else? Anything else? Moderna says, the chief CEO says it'll become an endemic disease and we'll be managing this forever. What's my opinion? Well, it's different than influenza. And the vaccines are much more effective than the typical influenza vaccines. So I think while we're getting variants, we can respond to them very quickly. But I think the other thing that's on the horizon is you're going to see a different kind of vaccine show up within the next 2 years. These are called the universal vaccines. They are not only antibody, they're T-cell vaccines. And they are geared to parts of the virus that can't change, because if they change, the virus dies. So there's going to be another generation of vaccines down the pike. And I think that coupled with the efficacy of these vaccines we talk about today, it's much less likely that COVID is going to become endemic like your seasonal flu. All right, we're getting close. We've got a few more minutes. If the RNA and adenoviral vaccines, DNA vaccines, are using full strength coding sequences of the spike protein, presumably the epitope presentation should be the same. Yeah, but if you're using adeno, you can't get the whole sequence in there. We've tried. In fact, that's why my company, we like Vaccinia as a vector. We don't like adeno. There's a limit to what you can pack in there. And the epitope presentation is not the same because each company has their own different consensus sequences that they are used to making their vaccine. It's not one size fits all. And as we saw, there's a lot of mutations in the spike region, and they vary from variant to variant. So why do you think there's a large efficacy difference between the variants and WT, the wild type? It's beyond the scope of this talk tonight to get into the mutants. I mean, we could teach a graduate course on that. How do the mutations, the specific mutations, correspond to biologic activity and resistance or non-resistance to a particular vaccine? But what it's showing us is that a small change, if it's in the right place, can have a dramatic effect. And if you look at the Brazilian and the South African strains, they have similar changes that are different than what you see in the UK strain or the other strains. So again, this small difference could really account for a lot of the differences in efficacy between the strains. You say J&J is the only one I know that's tested in South Africa. No, they tested the Novavax down there too. I can get amplification on that and exactly who went down there. But other vaccines beyond the AstraZeneca were tested in South Africa, and I believe Novavax was one of them. Could a COVID booster, if a COVID booster is recommended, do you need to continue with the same brand vaccine you have? That's a great question. I'm not sure if we got a solid answer to that. In Europe, they're studying that very question. Can you start off with one vaccine from one source and use your boosters and your variants from another? And so far, we don't have any data on that. All right. Andy, how are we doing?
I think maybe you got one or two more there and we'll do a closing. How about that?
All right, sounds good. Let me see if I can find any more. Is there a possibility of finding long-term negative effects of the vaccines, I presume? This is Pauline from Verona. Years from now. It's biology stuff is always possible, but I have not run into any vaccines that, with the one exception of that Guillain-Barré, that there's a delay in the onset of the symptoms of the vaccine response, and then they show up years. Guillain-Barré is a nasty case. It's basically a vaccine-induced demyelination of nerves. That is, the sheath that coats the nerve cord, the nerves, the afferent nerves. They're coated with these, they are coated with these proteins called myelin, and they are necessary for nerve conduction. And in Guillain-Barré, which is an induced autoimmune disease, you get demyelination, all sorts of nasty neurologic effects. Sometimes it's reversible, sometimes they're not. But as I said, other than in that 1976 swine flu, we haven't seen Guillain-Barré associated with any vaccine in recent years. And certainly it hasn't been described in any of the vaccines that we've been talking about today. But that's the only one I know of where there was a delayed response from a vaccine administration to a adverse event down the road. And so far, we haven't seen it. Does that mean it can't occur? Well, it's hard to predict a negative going forward, but so far it's unlikely. Real pharmacies are reportedly offering federal partnership Pfizer vaccines. Yes, in this state, CVS, as of tomorrow. The pharmacies are not yet equipped to maintain -70. Will they install dry ice freezers? Great question. I think they're going to rely on the shippers, which are dry ice shippers and do give you, a number of days that you can have the vaccine there to administer if you don't have a -70. You can bring them down to my laboratory. We've got about 6 -80s. We'll take care of them. But anyway, that's it. I think we'll cut it off there. If you have more questions and I didn't get to them, I apologize. What I think the best thing to do is, Andy, can they send them to you and then you can forward them on to me and we'll get an answer back?
Absolutely.
Yeah. Anyway, but again, I thank everyone for bearing with me. A lot of material. That's just unfortunately the way it is. And I hope I've at least been able to give you some information to make sense of it all, as I said. Again, thank you so much.
Special thank you to you, Dr. O'Connell. Thank you for your expertise. Thank you for volunteering your time. Thank you for your time that you put into preparing for this. And also thank you for your very thoughtful responses to excellent questions from all our participants.
Yes, really good questions today. Very nice.
I want to thank everybody for being part of this presentation. A month from today, we will be featuring David Alexander, who is on the Mount Olive Open Space Committee. We'll be talking about the environment. We'll be talking about the trails. We'll be talking about the actions of the Open Space Committee here in Mount Olive. For all the information regarding upcoming events, please visit the website, mountolivedemocrats.org. Feel free to sign up for our newsletter if you don't currently already get it. And again, I wish you safe, healthy, prosperous rest of 2021. Thank you again, everybody, and have a great night.
Good night, all. Thanks again.
Thanks. Bye.